Boreal toads were once common all over the Rocky Mountains and wetlands, but now that's no longer the case. Their populations, like thousands of other amphibian species around the world, have been devastated by the deadly chytrid fungus disease in recent decades. But things could be looking up.
Hi, sweetie. You are beautiful and large.
Dr. Valerie McKenzie, professor at the University of Colorado Boulder, and her team are working on a way to fight the fungus where it lives. Chytrid disrupts amphibian skin, which helps them regulate water, oxygen, and electrolytes, and spreads easily by swimming through water or even by direct contact between individuals. One of Mackenzie's colleagues recently discovered that certain bacteria that live on some amphibian skin, if isolated and cultured, can actually kill chytrid.
And that got me really intrigued because I thought, wow, I wonder if any of those types of special bacteria live on our amphibians.
So her team started culturing bacteria from toads in Colorado by swabbing their skin and growing the resident bacteria in petri dishes. They found one type, Xanthinobacterium lividum, or JLiv for short, killed the fungus.
So when we applied that to boreal toads in the lab, we saw a significant increase in their survival with the pathogen. And that was a great finding.
A 40% increase, actually. A massive success when compared to typical human drug trials. Now Morris Animal Foundation is funding the work to take what they've learned in the lab into the field.
How do we put this protective bacteria that's native to them here, how do we put more of it on them and get them to have a protected skin from this fungus in the wild?
And when? As adults? Eggs? This is a species that changes drastically as it ages. It seems that the magical time to treat them might be right after metamorphosis, just as their body and skin are changing from a tadpole state to a more formed toad.
That seems to be the critical window when we can treat them and get the treatment to remain successfully and have it persist and provide protection for the toads.
To test this theory, last summer they collected thousands of these metamorphs and soaked them in a probiotic solution of JLiv. The team has returned to multiple sites like this one in Buena Vista, Colorado, to see how it's working. They catch the metamorphs to swab them for samples to take back to the lab. So far, the results look promising. An incredibly important step to protect thousands of species so crucial to their ecosystems.
There's a good chance this will then be applicable to many other amphibian species, which there are lots that are being affected by this pathogen around the globe, and this kind of approach could work for other species in other places as well.
Making these once common toads common again.